lecture 3. And previously, we had some portion of the lecture 2, which is not taught. And this is the place that we have stopped last week. And let me try to persuade you that although I have introduced many species of bacteria, and you are not required to memorize the properties of these bacteria, and the only purpose is to introduce you to the multitude of this bacterial world. The purpose is that if we are able to know there are thousands and thousands of bacteria on Earth, and they play different roles in the biosphere, and in the future, you can study some bacteria in depth so as to produce a certain kind of application for your society or for the world economy. And currently, we are in the new trend of the genomics. This trend of genomics has the power to treat a lot of bacterial genomes simultaneously. So this is it's called microflora or microbiota. And having this ability to treat a lot of bacteria simultaneously in a certain niche. Niche means a place where they can live. It's called a niche, such as our intestine, our colon is a niche. And there are a few hundred kinds of bacteria in our colon. And we don't seem have the ability to process each one of them. Not to mention even to grow through every one of them. It's certainly not in the reach of our technology. However, we can sequence them. We can classify them. So this ability, this technology to treat microbiota helps create new directions in research. And so one of the more trendy research is to look at the animal gut microflora and to see how it is connected with health or disease or thin or fat. Some research showed that the microbiota can determine if you are fat or if you are thin. So these kind of techniques requires us to have a basic understanding of the current taxonomy or the current classification. So that's why I'm trying to introduce you to the whole scheme of classification. That is the whole rules scheme of 3 domains of life. So don't be frightened by so many kinds of bacteria. So you only need to look at the PPT or look at the textbook and to get some familiarity of some of those important ones. And gradually, with more effort put in microbiology, then you will be more familiar with the one which you are going to do the research. Okay, so let's pick up from where we have left off last week. That is the proteobacteria. Proteobacteria. It has 5 major branches of the proteobacteria. Proteobacteria means gram-negative, many-shaped bacteria, which by the phylogenetic analysis were put in a huge group. This group is called the proteobacteria. So it has 5 major branches. And I have introduced some of them in this phylum. Okay, let's continue. I have said that currently there are about 18 phyla of bacteria in this domain, and the biggest one is proteobacteria. But there are some others, which includes gram-negative bacteria, but phylogenetically distant from proteobacteria. So they are called gram-negative non-proteobacteria. So according to the dichotomous key, here we have the oxygenic phototrophs, which are the cyanobacteria, and anoxygenic phototroph, which are chloride. And these subsets of bacteria takes up sunlight, and they can use the energy from sunlight to synthesize their own cell bodies. But some can use oxygen, some cannot use oxygen. So these are the phototrophic ones, they're eating the light. And furthermore, we have other important bacteria. And so I do a little bit of introduction for you. Okay, let's see the next one, which is called the cyanobacteria. Cyanobacteria. This is important because these group of bacteria is able to release oxygen into their environment. That is, oxygen is their waste product. And just because they appeared about 3 billion years ago on Earth, so they gradually oxidize, non-oxidized, release the oxygen into the atmosphere, so as to convert a non-oxygen atmosphere into an atmosphere with about 20% oxygen in it. And their form, some are the filamentous beaded form, some are in globular form, some are in filaments. So they have, first, there are hundreds of species of cyanobacteria. Second, they are free living in the environment. And they have chlorophylls or other kind of pigment to catch sunlight and to produce organic material. Some have a certain kind of differentiation. That is, 1 in 10 of the cell will differentiate into a heterocyst. And this heterocyst is able to fix nitrogen. And this nitrogen is gradually diffused, is transported to its neighboring cells, so that the neighboring cells are able to fix carbon and to provide nutrient for heterocyst. And heterocyst fixes nitrogen into ammonia and is able to nourish the neighboring cells. So they are a form of mutualism. That is, they mutually help each other. Okay, so this is environmentally very important group of bacteria. Next, there are the purple, green photosynthetic bacteria. And some uses sulfur, some do not use the sulfur. And so they are called the purple or green sulfur bacteria. And some are called the purple or green non-sulfur bacteria. Some are really in proteobacteria. Some are out of the proteobacteria. But they are the major anoxygenic. Anoxygenic means that they do not take in oxygen as the final acceptor of electron pairs. Okay, so I've only mentioned this briefly, and then let me mention the later on I mentioned this spirochete. And first I'll try to explain this chlamydia. Chlamydia is jacketed, coated bacteria. Coated. Chlamydia is making with a bigger coat. They are very small, colloid, and gram-negative. And very strange group of bacteria. They are very small. They grow intracellularly. That is, out of cell, they are called elementary body. Once they get into the cell, they expand into a reticulate body. Reticulate means that we have many fibers in the cell body. Reticulate body is able to multiply. And then they differentiate into elementary body and shrink in size. And at this time, they will break the cell, kill the cell, and they will get out of the cell and be transmitted. And it is transmitted maybe by the droplet spilled out by coughing or by the respiration. So it became airborne or get into the feces. So in this way, that is, if people are inhaling the feces of birds, or because of the interpersonal contact, this chlamydia can transmit to other persons. So these are very important pathogens. In the human being, it is called trachoma. Trachoma is sandy eyes. That is, they will form granules on the eyelid, and always on the inside of on the surface of the cornea. Because they are granules, it's like a sand stuck in people's eyes, and this sandy granule will scratch the cornea, so people will get blind due to the growth of this bacteria. But this bacteria can also be transmitted to sexual organs. And in the sexual organs, it became a sexually transmitted disease, and it can have very severe sexually disease syndrome, such as a wart or a lesion on the outer sexual organs, and sometimes something which are terrible to look at. So this is not only going to the eyes, it also goes to the sexual organs. So this is a very important STI of a neural disease called chlamydia trachomatis. And some are in birds. It's called psittacosis. Psittacosis means psittaco means parrot. The Greek name for parrot is psittacos. Psittacosis is called bird disease, bird fever, because in its feces, there are chlamydia bacteria. So if you grow pigeons, you as pet, the feces of pigeon if you inhale, you produce the respiratory disease, pneumonia. So this chlamydia is very tiny intracellular parasites, which can cause severe human diseases as well as bird diseases. So this is chlamydia. We introduced some interesting about chlamydia. Then bacteroiditis. Let me show you this one. Bacteroiditis. This one is interesting because it's anaerobic. It's a bacillus, and it's the most abundant bacteria in human large intestine. That is, its density can go up to 10 to the 11th power. 10 to the 11th power. That is a trillion. A trillion is a billion, right? Billion is a billion is 10 to the 9th. So this is 100 billion cells per gram of intestinal material, and it cannot grow in the presence of air, and it's the most abundant in the feces. And we really don't know its function in our human large intestine. However, it is the most abundant ones, and it's gram negative and anaerobic and non-motile. That is, they cannot move. That is in contrast to E. coli, which can move, which is also a constituent in our human large intestine. But the number of E. coli is much smaller than Bacteroidetes. Okay, so this is an important bacteria to explore. And furthermore, this is Fusobacteria. Fusobacteria is a spindle-shaped bacteria. It's gram-negative, long and slender in the human gums, That is, in the teeth, underneath the teeth. So it may cause the dental diseases. Okay. Then there are a few other phylum in the bacteria domain, which are quite interesting. This one, Thermus, Deinococcus and Thermus belongs to this phylum. And Thermus is important for its ability to grow in very hot environment. It's called the Thermus aquaticus. It's able to grow in the hot spring of Yellow Spring National Park. This species was isolated by Brock, Thomas Brock, who is a pioneer in the hot water microbiology. So he isolated, he was a professor at the University of Wisconsin, and it was near the Yellowstone National Park. So he went there and made a lot of collections from the hot springs. Hot springs temperature can be as high as from 80 to 95 or 97, 98. And he's able to observe string-like, fiber-like material, slimy things. So he thought that this may be bacteria which can grow in hot water. So he isolated many, and then he was able to grow them, to purify them. And one of those is called the Thermus. And in 1986, when PCR reaction was invented by Mullers, some people thought, DNA polymerase from the thermophile bacteria may not be killed by heating, so they try to purify the DNA polymerase from Thermus aquaticus. They are successful, and so this DNA polymerase, as well as the later brand of heat-resistant DNA polymerase, became a whole industry. Its sale can be $20 or $30 billion worldwide. So this is industrially very important bacteria because of its application in the PCR reaction. And this Deinococcus is Dino means terrible, horrifying. Deinococcus is Dicoccus, Diplococcus, or Tetracoccus. The major property of this Deinococcus is that it can withstand the radiation up to 150 times, another 100, not 1500 times, 150 times the dosage that will kill a person. That is, if we give it 1500 grays of alpha or gamma radiation, then they will not be killed. And they have very strong DNA repair to repair the damage to the DNA. Generally, humans cannot withstand more than about 50 grays of radiation. That is in a nuclear power plant breakdown. People, if they are exposed to more than 50 grays, will die eventually. Okay, so this is a small branch of small phylum in the bacteria domain. Let's go back to the two previous ones. I have not talked about the spirochete. So later on, I will talk about spirochete, which is also gram-negative bacteria. But let's first go into the gram-positive bacteria. Gram-positive bacteria is one single phylum, phylogenetically somewhat related to gram-negative bacteria, proteobacteria, but it's separate, and so they are different phyla. However, this gram-positive bacteria has many important pathogenic bacteria in it. So I introduced some important ones. First, they are divided into two branches. That is low GC ratio, and the second one is high GC ratio. For the Streptomyces, which has the highest GC ratio, and Mycobacterium, the second highest ones. And these low GC ratio ones are from 40 down to about 21, 40 or 50 down to 21. So these are the important ones. So they are called Firmicutes. Firm means strong, hard. Cutis means skin, hard skin. Hard skin means denote its peptidoglycan. That is, it's abundant type of peptidoglycan. Also describes them as gram-positive ones. Actinobacteria, actino is shooting out, like a fungi shooting out. That is, they are filamentous and shooting out many branches called actinobracteater. So all these are collectively called actinobacteria, and all these are collectively called Firmicutes. So these are important in the microflora literature. Okay, first is the low GC ones. Low GC ones, we can see there are four major branches. This one is Mycoplasma. Mycoplasma is one human pathogen. And the Clostridia, spindle-shaped gram-positive ones. The major properties are anaerobic. The bacillus are large bacillus form of gram-positive bacteria. And then the Lactobacilli lalis is without endospores. So these four categories of gram-positive low GC ones are all quite important. First, the Mycoplasma. Mycoplasma is interesting. That is, they do not have cell wall. They do not have cell wall, so they do not have peptidoglycan. But how can they be classified into this category? It is because they are 16S RNA, and first they are small, and then they can grow in human lungs, and they can also grow in tissue culture. And they can grow in tissue culture as a very small colony, like a fried egg. That is the center is raised, and the periphery is flat, like a fried egg, and it can cause mild pneumonia in human beings, and so this is very interesting. Okay, then let's go into the Clostridia. Clostridia means it's a fusiform, spindle form, the bacteria. And the important ones are called Clostridium, and the major properties are its endospore. Some endospore are located at the terminal. It's called terminal endospores. Some endospore are in the middle. It's called medium endospores. So using the position of endospore, we can distinguish different species of Clostridium. And medically, there are three kinds of Clostridium which are important, and I only mentioned two. First is botulinum, Clostridium botulinum. Botulinum means sausage. Botunus means sausage. And if the sausage is not heated well, then this bacterium spores may get into the meat. And then, because it's a sausage, it doesn't have oxygen, so the bacteria will grow and they release this toxin. And so this will cause very fatal disease. That is, the poisoning is very fast, and the people will die in a very short time. And in each culture where people make sausages, there are some death each year due to eating of the poisoned or bacteria-laden sausages. And this toxin is called the botunus toxin. Botunus toxin is able to relax the muscle. So it was changed into medicine called Botox. Botox is when it is injected onto the face, then it will make the wrinkle disappear. So it became a cosmetically important drug. This tetanus releases toxin, which will cause spasm. That is the shrinking up, the cramping up of the muscle. Botunus toxin will release the muscle, but the tetanus toxin will that is clamp up the muscle, and so people will die very horrifying death because the muscle in the back, or the muscle in the lung, or the muscle for the breathing, are all in spasm. So the death is terrible. So it is a tetanus. And the toxin, we call these toxins the AB toxin. AB toxin means that it has two parts. One part is to get into the cell. The second part is to kill the cell. So they have the make a toxin, and these toxins are called exotoxin, which are just different to the endotoxin. And I have told you that endotoxin is the toxin which is LPS, lipopolysaccharide. That is a part of the bacterial outer membrane, which when released into the animal bloodstream will cause a severe shock, bacterial shock, which will kill the animal or the human beings. And the toxin made by bacteria is called exotoxin. That's specifically made by the bacteria to release into the animal, which will kill the animal. So this is Clostridium, and then I introduce a little bit about the Bacillus, and when we see Alice, this is a family. The status is a family. This Bacillus is very important in terms of the warfare. Okay, the original one is Bacillus anthracis, which is causing the black legion on the surface of the animal, so kill cattle, sheep, goat, and human beings. And the important ones are some strains of Bacillus anthrax, which will generate spores, and this spore can be inhaled by human beings, get into the lung. And when it gets into the lung, it's very pathogenic. That is, the human will die very quickly. And so this is one of the most important biological weapons, or the terrorist weapons in the world. That is, some organization will be able to grow bacillus and then make them into endospore form. This endospore form can be stored hundreds of thousands of years. This endospore will be be sent out through the envelope, or through a bomb, or through some explosive, and so when it gets into the human lung, then it will kill people quickly. So this is important in terms of the military. Okay, biological weapons. And Staphylococcus is also gram-positive ones, gram-positive bacteria. Okay, and let me mention a little bit of Lactobacillus. Lactobacillus is aerotolerant anaerobes. That is, it is able to grow without oxygen, but in the presence of oxygen, it will not be killed. So the important ones for the food industry is Lactobacillus. It can make all sorts of preserve all sorts of food, especially vegetable. So in Korea it's called kimchi, and in China it's called Sichuan, preserved vegetable, all made by the Lactobacillus. And also for making the yogurt or pickles, all sorts of sour food. And this is very important. And Streptococcus is a linear form coccus, And this is important human pathogen. It will release the pathogen called streptomycin, which will lyse the blood. And so at the same time will cause a lot of diseases, such as this one, Streptococcus pyogenes will cause strep throat, cause scarlet fever, rheumatic fever. So a whole group of diseases may be caused by this Streptococcus. So this is Streptococcus pyogenes. And the Streptococcus pneumoniae is the agent of bacterial pneumonia. Bacterial pneumonia is the more severe form of pneumonia, which is also the final reason of death for elderly. Because when they get influenza, and then influenza will cause this bacterial pneumonia, and bacterial pneumonia will very fast cause the collapse of the human beings. Okay, in gram high GC ratio gram-positive ones, it's called actinobacterial. Actinobacterial ones, there are many important ones. So I mention only a few important ones in actinobacteria, such as Corynebacterium. Okay, let me first. If they form filaments, some are called nocardia, some streptomyces. Okay, let me show you. Okay, important actinobacteria. Here are the 6 important ones, which I introduced a little bit. The important bacteria here is Corynebacterium, which was called diphtheria. Diphtheria is a whitish membrane in human throat, which is a disease in human childhood and cause death very quickly because it has a diphtheria toxin. And this Propionibacterium produces a propionic acid, which is the major bacteria for making the Swiss cheese. Mycobacterium is the major pathogen for tuberculosis. So this is the one which was studied by Koch, Robert Koch, which made a tremendous contribution to medical microbiology because tuberculosis was confirmed to be a bacterial disease. Leprae is causing the leprosy. Leprosy is a terrible disease which deforms the human face, deforms the bones, the human hands and feet, which in the biblical time was terrifying people. So they gathered all the leprotic persons into a place, that is where they were exiled into that place, not allowed to back into the human society. And this streptomyces, strepto means long filament. Streptomyces is like fungi, especially has mycelial growth. Mycelial growth and has an earthy odor. That is, if you will smell the earth, then the smell of the earth generally came from the streptomyces. It produces lots of antibiotics, more than 500 kinds of antibiotics. Most of them are used for research because their toxicity is too high for human use. Only a few were used for human disease, such as streptomycin. So these are their importance. We can see that they form spores. And these are spores of very different forms. So they look like mold, but they are not mold. Mold is fungus, but this streptomyces is like looking like mold, but they are bacteria. And let me summarize this one. Finally, give you some introduction to the Lyme disease and also spirochete. Spirochete is spiroform, spirillum, spiroform, the bacteria. They may have the very tight coil or very loose coil. In terms of tight coil, the pathogen of syphilis, it's called the Treponema pallidum, is one form of spirochete that is a tight coil. This syphilis is one very famous venereal disease in human beings. And since Columbus went out to explore the world, when he came back to Europe, especially Spain, and the syphilis appeared in Europe and then spread out to the whole of Europe. So people call it the Spanish disease, or some people call it the English disease, or call it the French disease. And the syphilis eventually get to the Orient, and so start spreading from the Macau, to the Japan, to China. And so apparently this is a disease of the New World, was carried back by the sailors from the Central America back to Europe, and then eventually to Asia. So it causes excruciating disease for human beings. So they are the first phase of disease in the sexual organs, and the second phase is throughout the whole body, and the third phase of the syphilis is getting into the brain. So the people will get a psychotic. So this is a terrible sexually transmitted disease. It's called the syphilis. And the Lyme disease is caused by Borrelia. This Borrelia is a loose spiral. Although it's a thin bacteria, but it's a loose spiral. This is spread by ticks. Ticks is a kind of parasite, like a small parasite, which will drink blood from animals. This tick generally lives on the body of deer, and these deers, when they in the springtime, they may invade the forest, and those forests may be near human families. So children may play in their garden, which has this tick in contaminated with this Borrelia, and so this tick bite causes this Lyme disease. Lyme disease will cause red ring, red lesion on the skin, and this red lesion will spread out, gradually spread from one red spot to a red ring, and sometimes cause the joint pains. So this disease happens in Lyme, city Lyme in Connecticut, United States. So it's called the Lyme disease in the 1980s. And then it was a sporadic disease in the rural area of America. So this is a newly appeared disease. Okay, so I have used 1 hour, that is 50 minutes, and to introduce the various important species in domain bacteria. So the purpose is not for you to memorize it. That is the only read it and get knowledge from it, and then gradually absorb the knowledges about each different bacteria. So this content will not be tested. So don't worry, they will not be tested because they are two tidbits. Like so, it's not. I will not demand to you to memorize it. However, it is important in your research and also important in the current direction of microbiological research. Okay, so I have rest for you for 10 minutes. So we'll start at 9:25. 9:25.